Okay, let's start. Good morning, afternoon, evening. Hola, hello, bonjour, hola. Ni hao, konnichiwa, 안녕하세요. And hello all. And welcome and thank you, SynBYSS people, for joining us, the 83rd seminar, and I call it on UC Berkeley Day. I am in Los Angeles, very close to, today's the pioneer speaker, Doc, attending SEED conference. It is so great to see many friends, colleagues, and young researchers all over the world to discuss science and engineering. I am very grateful for such conferences and overcoming the pandemic almost. We always take important things for granted until we lose them. Okay, it is my tremendous honor to introduce very briefly, I'm not sure I can do so, our pioneer speaker, Professor Doug Clark. He does not need any introduction because we all know him. Also, it would be almost impossible, as I said earlier, to briefly introduce him. He is a dean of UC Berkeley, a national academic member of engineering, and the editor-in-chief of biotechnology and bioengineering. I had an honor to receive a BNB Daniel Wang Award, he probably mentioned briefly later, given by the journal several years ago. At that time, he gave several minutes of introduction. I still remember. But I just need to briefly introduce today him. So Doc, I truly thank you for your time and for your contribution to the scientific society, especially education of younger generation, despite your super busy schedule. And it's my honor to have you today, and the virtual podium is all yours now. Thank you again. Thank you.
Well, thank you, Tae-sok, a very kind introduction, and it's a real honor for me to be here. Great pleasure, great pleasure. So let me start by sharing my screen and showing just a few slides. Now, this will just take a moment. My screen turns blank for a moment, but it will soon reappear. And there it is. So Tae-sok mentioned that I'm the editor-in-chief of biotechnology and bioengineering, and I first met Tae-sok in 2019 when, as he said, he was the winner of the BNB Daniel I.C. Wang Award. So we recognized Tae-sok's excellence then, and it's great to see that the arc of his career has just continued to rise and that he is doing so exceptionally well. So it's a great pleasure to be a part of this program. And when Tae-sok asked me to just say a few words, I, of course, was quite honored and asked him, well, what is it that you would like me to say? And what would you like me to talk about? And his response was shown here. You can say anything that inspires young researchers, including your vision, perspectives, and insights into the field, as well as career advice. So that covers a lot of ground. I don't know that I can offer too much in the way of insights and career advice, but I'll try to offer a few helpful remarks. So let's turn the clock back to when I was still in graduate school, and the year was 1983. And my advisor was Jay Bailey, who was shown here, who was a true pioneer in the field. And pictured here is Elmer Gaydon, another pioneer in the field, and the editor-in-chief at the time of the journal Biotechnology and Bioengineering. This which we'd submitted my very first research paper back in 1983. And here is the cover letter that Elmer wrote by hand. He wrote all of his correspondence by hand. It was a different time then, and sent to Jay with a single review of the manuscript. Now, why am I showing you this? And for that matter, why did I save this? Well, I saved it because it meant a lot to me at the time, and it still does. I had great respect and admiration for both Jay Bailey and Elmer Gaden, and I still draw inspiration from the positive feedback that this letter represents. The letter also illustrates how much the field has changed since then. So notice the reference number in the upper right corner, 83123. That means that in July of 1983, halfway through the year, 123 papers had been submitted to B&B, which at the time was essentially the only biotechnology journal around. We now receive over 1,200 submissions per year, and as all of you know, there are many other biotechnology journals to choose from. So obviously the field has grown tremendously in size and scope since 1983. And even the cover of the journal has changed dramatically. Here it was in 1983, and here it is in 2023. Definitely a change for the better, I'd say. And if we consider growth of the field from 1983 to 2009, which marked the 50th anniversary of the journal, not that long ago, here was a list of topics encompassed by the journal and the field 14 years ago. I won't read through this, but you can see it's a very extensive and comprehensive list of different subtopics within this very broad field of biotechnology and biochemical engineering. This list is still fairly up to date, and now, of course, we're all anxious to see what AI has to add and how it will revolutionize the field, if in fact it does revolutionize the field. So this all leads me to just a few not so deep thoughts. So let me summarize just a few perspectives that I can offer. There has never been a more exciting time to be working in the field of biotechnology. And there has never been a more important time to be a biochemical, a biomolecular, a bioengineer, given the problems that the world is facing and the contributions that biochemical engineering can make towards solving those problems. So for young investigators out there, choose problems that truly interest and inspire you. They will be the most fun to work on, and they will be the problems that you can have the greatest impact toward. And draw inspiration from whatever and whomever you can. For me, I draw inspiration from a letter that was written decades ago. And I still draw inspiration from many other sources, of course, but wherever, whatever inspires you. And savor the successful moments along the way. The acceptance of your first research paper, for example. That's an important milestone. And learn all you can from the failures. And then, as a very wise person once told me, if it isn't fun, something isn't right. So whatever you're doing, you should be enjoying it. So those are just a few thoughts that I can offer to young scientists and researchers out there. And as Elmer Gaden himself once told me when I interviewed him for a video that you can see still on YouTube at the end of the interview, he said, "That's all the wisdom I got." Well, that's all the wisdom I got. So let me again thank you, Tae-sok. It's been an honor to be a part of this, and congratulations to Sangmoo for being today's invited speaker as part of this great program. So thank you. I'll stop.
Oh my God, thank you so much. That, oh my God, that resonates with me a lot. Oh my God. So, you know what, my first paper I submitted, but at the end of the day, we couldn't publish it. At that time, we also print out 3 copies and then by surface mail, and then we sent to that. But we wanted to submit to the B&B, but the quality of the paper was not sufficient enough, so we decided to go to another journal. That was the 20, oh my God, that is already 26 or 5 years ago, something like that. So yeah, that was one of the best journal at the time. B&B was the best journal at the time, and we could not submit our paper. My first paper at the time. So you refresh my memory. And also you also mentioned this is the best time, also wonderful time to be biotechnology researchers. I completely agree with you. I also switched my field from applied chemist. That's my bachelor degree. And you mentioned Jay Bailey. Actually, Jay Bailey's textbook I took as a chemistry student. I just wanted to know what biology looks like. And that textbook basically transformed me from chemist into biochemical engineer. So it's my super honor to have you again because you are the, big fathers, basically biochemical engineering fathers kind of academic son. So it's my great honor to have you again. And Sangmoo also changed his field, so that also probably resonate with him, I guess. So that absolutely brilliant talk from you. Thank you so much.
My great pleasure. Thank you.
It was really short. I wish I would listen to you talk longer, but yeah, next time.
Maybe next time. Now it's time to hear from you, Sangmoo. That's why we're all here.
Thank you so much, in talk again.
You.
So this is absolutely wonderful presentation. Thank you.
You're very welcome. Thank you.
Okay, so now the main speaker of today with the longer introduction. Dr. Sangmoo Jeong is an assistant professor in the Department of Chemical and Biomolecular Engineering at Johns Hopkins University. His lab is interested in how metabolic dysfunctions link to major diseases, including cancer and inflammatory bowel diseases. I'm very interested in this kind of disease myself. With expertise in device engineering, as I mentioned, he switched field completely from those device engineering to biology. His lab focused on developing technologies to deepen our understanding of local and systematic metabolic malfunctions in diseases and identify how our daily diet affects disease progression. He received a Bachelor of Science degree in Electrical Engineering from the KAIST in Korea and a Master of Science and PhD degree again in Electrical Engineering from Stanford University. He completed his postdoctoral research, that is basically transition point for him, at Memorial Sloan Kettering Cancer Center. During that time, he developed a micro NMR system to quantify metabolic flux in cancer stem cells. He is a recipient of the NIH K99, ROO, and MIRAC Awards, one of the most competitive NIH grants. Sangmoo is a truly rising star with a huge potential to become a leader in the future. So I'm so excited to listen to his talk today. And if audience, one of you, let's say you are the big name, potentially writing a tenure letter, right? Tenure letter, I strongly, recommend you should write your strongest letter for him.
Oh, thank you.
Sangmoo, thanks so much for your time today. And please take it away. Thank you.
Okay, thank you very much. It's a really great introduction. So, okay, I'm excited to be here, not only because I can share my research, but also I can advertise my labs, because we are expanding now and I can discuss more later. So as Dr. Moon mentioned, my lab investigates metabolism. So in my opinion, metabolism is the most fundamental biochemical reactions that regulates every single piece of our living. So my lab investigates how these metabolic dysfunctions are connected to major diseases such as cancer. And as an engineer, we are developing metabolic biomarkers and sensing technology. So I started my lab in 2021 in the middle of pandemic. Yeah, it was full of very memorable and challenging moments. And so for the last 2 years, my lab has established 3 research directions. So first one is we are trying to understand how cancer cells change their metabolism along the metastasis processes. So people have now realized that cancer cells have very different metabolic features between primary tumor sites or during circulation and metastatic sites. So if you understand how they change the metabolic features along the path, maybe we can target them to minimize or inhibit the metastatic process. The second direction that we are focusing now is to engineer probiotics for more effective delivery and treatment for bowel disease or colon cancers. So as you may know, only a small portion of probiotics could survive in the gastric fluid and reach to your gut. So our labs develops novel coating materials for more effective delivery of probiotics. And we also plan to engineer metabolism of probiotics for generating biomaterials favorable to our gut health once it reaches the gut. And the last research direction is the metabolic engineering of mesenchymal stem cells, especially extracellular vesicle generations. So these vesicles from mesenchymal stem cells can be used as therapeutic agents, but the limitation is the generation of stem cells. So we aim to engineer the metabolism of mesenchymal stem cells so that they could massively generate therapeutic EVs while minimizing being differentiated. So if your research interests and expertise fit well to these 3 topics, please contact me. I'm expanding my lab and we are hiring postdocs. And these 2 topics are kind of early phase from our lab. So today I'll share mostly our research from this first topic. So let's get into that about cancer metabolism. So as many of you are well aware that the glucose concentration from the culture media is way over the physiological, pathological levels. So, excuse me. So, but actually the tumor microenvironment glucose level is, some people say less than 2 or 3 millimolars. And people have suggested that it's possible that cancer cells can use lactate or other metabolites for their major carbon sources. And actually, a couple of people have recently pointed out that cancer cells can use fructose, which is a sweet sugar, as a major carbon source. So, especially about 7 years ago, there's one paper published in Cancer Cells showing that acute myeloid leukemia patients, AML patients, have very high level of fructose in their bone marrow. So please keep in mind that the bone marrow for leukemia patients is the tumor microenvironment where leukemic cells are generated and propagated through your body. And the normal fructose levels in healthy patients, healthy people, is less than 0.5 millimolars. And somehow, leukemic patients have over 2 millimolar or 3 up to 5 millimolar of fructose in their bone marrow. And this paper suggested that leukemic cells in the bone marrow might use fructose when the glucose becomes limited. So the mechanism was not that clear, and I was kind of jumped into this topics during my postdoctoral research. So before I go deeper into the fructose metabolism, let's briefly talk about our dietary fructose consumption. So from Western diet, it's really hard to avoid fructose consumption. So if you drink Coke or if you have maple syrup with your pancakes, and if you put the sugar on your coffee, you end up having fructose. So mostly these syrup and sugars are form of sucrose, and this sucrose can be cleaved and observed separately as a glucose and fructose in the small intestine. And if you see the fructose metabolism a little more deeper, it makes sense that cancer cells can use fructose for their major carbon sources, because fructose can be metabolized through the KHK or hexokinase, and especially intestine and liver, fructose is metabolized by KHK and becomes fructose 1-phosphate and converted to other metabolites. But other organs, they normally metabolize fructose through the hexokinase, and then fructose becomes fructose 6-phosphate and joins the glycolytic pathway. So if the glucose becomes limited, it's possible that cancer cells use fructose to generate glycolytic intermediates and then continue their proliferations. So I started culturing the leukemic cells, like 4 different types of acute myeloid leukemia cells in 3 different conditions of media. The first is regular glucose-rich media, RPMI. And second media is I completely remove glucose, but I add fructose. And third one is there's no glucose and fructose. In the third case, as you see in the green line, cells couldn't survive at all. But interestingly, cells grow absolutely fine whether they have glucose or fructose. And especially one cell line grew better when they're fed with fructose. And when we analyze their metabolic consumption, we found that fructose consumption was significantly lower than glucose consumption. And it's not surprising that the cells secrete way less lactate in the fructose condition because they consume less amount of carbon. And we found that the leukemic cells in the fructose-rich condition, they consume more glutamine, which is expected because it has been known that when the cancer cells have limited glucose, they upregulate glutamine metabolism to populate all other metabolites, intermediates. And in the fructose-rich condition, as you see here, the consumption of carbons is much lower than glucose environment. So they have to use more glutamine to achieve similar or better proliferation. It makes sense, and it's not that surprising. But surprising feature comes from this slide. So when we culture the cells in carbon-2 labeled glucose or fructose, we found that carbon-2 glycine NMR peak was extremely high only in fructose case. So when we do unbiased metabolic analysis, we found that carbon-2 glycine, carbon-2 serum levels extremely higher only in the fructose-rich conditions. So this is the metabolites in the media. So basically, they secrete out serine and glycine. And we also analyze intracellular level of metabolites using mass spec. And as you see here, the total pool size of serine and glycine levels much higher in the fructose condition. And when we do fractional enrichment analysis, the cells have higher enrichment of serine and glycine when they are cultured in fructose-rich conditions. What does that mean? It means that leukemic cells upregulate the serine synthesis pathway in the fructose-rich condition because labeled serine and glycine generation only possible through this pathway. So when you feed the leukemic cells with carbon-2 labeled glucose, the cells can make carbon-2 labeled serine and glycine, or carbon-2 labeled fructose, which can generate carbon-2 labeled serine and glycine. So I started looking up more details about SSP, or serine synthesis pathway. And actually, this pathway has emerged as a key pathway for cancer because the end product of serine or glycine, it can be used as to generate the precursor to generate a glutathione, which is a major antioxidant. And these are precursors for nucleotide synthesis and lipid synthesis. And this pathway also generates alpha-ketoglutarate, which is a TCA anaphylaxis precursors. So this pathway is extremely important for cell proliferation, and subset of cancer cells upregulate the rate-limiting enzyme of this pathway, PHDH. So we started investigate the role of PHDH on the leukemia cells metabolism. So when we knocked down PHDH with doxycycline, we found that the carbon-2 labeled glycine peak was gone in the fructose-rich condition, which means that the leukemic cells generate glycine from the SSP. It's confirmed our hypothesis. And we compare the proliferation rate between glucose and fructose condition with knockdown of PHDH, as you shown here. Even glucose condition, the leukemic cells lose their proliferation when we knock down PHDH, but the effect of PHDH inhibition was more significant in the fructose-rich conditions, which means that leukemic cells are more dependent on fructose, SSP in the fructose conditions. Okay, this is all about leukemia cell lines. So we tested their normal counterparts, which is normal CD34 positive hematopoietic stem cell rich populations. Interestingly, these normal population or normal hematopoietic stem cell rich populations, they don't upregulate SSP. So their metabolism was similar between glucose condition and fructose condition. And when we inhibit the cells with PHDH inhibitor, they don't respond almost at all. And as again, the leukemic cells, their growth rate decreased significantly with the PHDH inhibitor in the presence of fructose, but normal hematopoietic stem cells, they don't respond at all, which means that this SSP targeting can be an effective treatment option for leukemia. So we tested, we was able to get the leukemic patient samples, and we culture them in glucose and fructose condition, and we measure the apoptotic biomarker, apoptotic levels. And again, with the PHDH inhibitor treatment, of course, they show higher apoptotic markers in the glucose media, but in the presence of fructose, in the absence of glucose, their apoptotic levels was significantly higher with the PHDH inhibitors, which means that, again, leukemic cells, cell line, as well as primary leukemic cells depend on SSP or serine synthesis pathway in the fructose-rich condition. Okay, the next question would be, why leukemic cells upregulate this SSP in the fructose-rich conditions? And we found that the cofactors NAD plus and NADH levels are significantly different between these two conditions. And the PHDH, the rate-limiting enzyme in this pathway, use NAD plus for their reaction. And in the fructose conditions, the ratio of NAD plus and NADH was much higher, which facilitate this SSP reactions. And we play around the ratios by adding lactate, supplementing lactate or pyruvate. So when you add lactate into the culture media, cells take up lactate, and then the LDH enzyme generate pyruvate by consuming NAD+. So it's very quick reactions. So when we add lactate into culture media in the presence of fructose, their NAD+ over NADH level ratios decrease significantly, which kind of stops the SSP reactions. Because basically, we couldn't see any glycine generation from the leukemic cells in the fructose-rich media. And on the other hand, when we add pyruvate in the culture media, cells take up extra pyruvate from the media and then consume NADH and generate NAD+. So when we add pyruvate, the ratio was increased, which provides more precursors for PHDH reaction, and we confirmed that glycine generation was increased even in the glucose conditions, glucose-rich conditions. So we confirmed that this NAD plus NADH ratio determines the metabolic flux through the SSP because PHDH enzyme use these cofactors. And I don't go deeper, but we also found that by upregulating upregulating this pathway, leukemic cells can generate more alpha-ketoglutarate, which provides TCA anaplerotic reactions. So overall, these metabolic advantages support leukemic cells' growth in fructose-rich environment. And we also tested in vivo models. So we make the leukemia mice, and we injected glucose or fructose with PHDH inhibitor. We confirmed that enrichment of glycine was significantly increased when we add fructose, which kind of confirming our in vitro findings. So lastly, I want to share one really important, interesting observations. So as I mentioned before, the researcher found that leukemic patients have high level of fructose in the patient's bone marrow. It was not known why. And also we found that leukemic mice, their fructose level was higher in the bone marrow. Of course, glucose level is lower because the leukemic cells in the bone marrow consume glucose, but still we couldn't figure it out why fructose level is higher in the leukemic bone marrow. We are working on now, and if you have any ideas and insights, please let me know. So with that, I can wrap up my first part of the research. So basically, we are showing that leukemic cells are very able to change their metabolism based on metabolic conditions, and they are able to uptake fructose when glucose becomes limited. And in that condition, they upregulate this SSP to support proliferations. Okay, so let me switch gears slightly to breast cancer metabolism, metastasis, because there is a connection between leukemia cell metabolism and breast cancer metastasis. Maybe some of you have heard about E-cadherin, which is adherent junction molecules between the cells. And when the cells are tightly packed, the levels are upregulated, as you show in this schematic. And because of this kind of cell-to-cell adherence junction molecules, if higher expression of E-cadherin was thought that it's a tumor suppressor. Because people show that when the cancer cells lose the expression of E-cadherin, they start migrating to the other organs. It makes sense, right? If higher expression of E-cadherin, cells tend to attach each other and they don't leave from the primary site. And once they lose this adherent junction molecules, they start migrating and then go to another site. But recently, people have suggested that probably E-cadherin loss is not required because cells form as cluster for their metastasis, like as you see in this schematics. Some portion of cells might lose E-cadherin, but overall, the loss of E-cadherin is not required for metastasis. And this is kind of multiple papers I have shown this research. Of course, it depends on cancer types, and there's no universal rule or consistent observations about the role of E-cadherin. But anyway, recent papers demonstrate that E-cadherin expression helps the metastasis as well as primary tumor growth. So E-cadherin-positive breast cancer cells grow much faster here, and E-cadherin-positive breast cancer cells metastasize more to other organs. So hyperproliferation or higher metastasis actually requires specific metabolic reprogramming because the cancer cells needs more precursors for biosynthetic processes. Also, they have generate more glutathione to minimize the RS or oxidative stress during metastasis. But it has not been known which metabolic pathways are connected to E-cadherin. And from this bilateral tumor models, we found that E-cadherin high positive tumors upregulate PHDH and PSET1. So these enzymes, actually, I briefly mentioned that, is a key enzymes in this SSP, serine synthesis pathway. Again, leukemic cells upregulate this pathway when glucose becomes limited. And breast cancer cells, E-cadherin positive cells upregulate these metabolic enzymes. And we do more studies, and we found that the E-cadherin positive cells upregulate this glycine serine metabolism, which is validated by the 3 cell lines. So MDA MB231 breast cancer cells, they are intrinsically, their level of E-cadherin is low. So we overexpress E-cadherin, and we found that PHDH, PSET1 level is also upregulated. And these two breast cancer cell lines, their E-cadherin levels intrinsically high. So in these cases, we knock down E-cadherin, and again, PHDH P7 levels decrease as E-cadherin levels, and as well as the mRNA levels. It's very interesting. Why E-cadherin is linked to PHDH or P7, we don't know yet. So we also did a metabolic flux analysis using a mass spec. So when we culture the cells at fully labeled glucose, we found that the fully labeled serine and glycine generation is significantly higher in ECAD positive breast cancer cells. Okay, so which means that they are dependent on SSP for their metabolism. And when we knock down PHDH, yeah, their growth rate was hampered and becomes similar to E-cadherin negative cells. And E-cadherin negative cells don't respond to PHDH inhibition as well as PHDH inhibitor treatment. Again, it clearly indicates that E-cadherin positive cells are dependent on PHDH or SSP for their proliferation. Also, E-cadherin positive cells, their RS levels increase significantly when we inhibit the PHDH. So again, E-cadherin negative cells don't respond at all in terms of RS, but E-cadherin positive cells, two cell lines, their RS levels was significantly elevated when we treat them with PHDH inhibitors. And again, so this is a basic level of RS. So we did one step further. So we apply some kind of acute exogenous RS stress by adding hydrogen peroxide into the culture media. So when we add hydrogen peroxide, definitely RS levels went up in all the cell lines, all the clones. But when we knock down PHDH, the elevation of RS was much significant under this acute exogenous RS stress, which means that, again, ECAD positive cells have higher RS reducing capacity, but if you inhibit PHDH, they lose their metabolic advantage. And in vivo experiments, we found that, yeah, ECAD positive cells grow much faster than ECAD negative tumors, but only ECAD positive tumors respond to PHDH inhibitors. Okay, this is primary tumor growth. And we also tested metastasis. So basically, we primed the cells with PHDH inhibitor versus vehicle for 24 hours. Also, of course, after 24-hour treatment, cells don't die at all. And after that, we injected the mice through the tail vein. And after 48 hours, we checked the metastasis into the lungs. Basically, it's we're trying to see metastatic potential regardless of primary tumors. Because in this case, we also saw much higher metastasis to the lungs in the ECAD positive vehicle treatment groups, but it is due to the size difference of primary tumors. But in this case, we injected the same number of cells and just analyze the metastasis, micrometastasis to the lungs. And as you see this red dots, the breast cancer cells treated with vehicles were able to metastasize to the lung much efficiently compared to the PHDH inhibitor treatment. Okay, so like this part, I explained that ECAD positive breast cancer cells, they upregulate SSP, which is beneficial for them for proliferation and for RS management. And previously, I talked about leukemia cells. They upregulate SSP to generate metabolic precursors for growth in the fructose-rich condition. But ECAD positive breast cancer cells, they upregulate this pathway for their proliferation as well as RS management. So we found that this PHDH inhibition hampers private tumor growth as well as lung metastasis. So with that, I can stop now. My lab has been about 2.5 years in Hopkins, and thankfully I have great collaborators and generous support from the funding sources. And I can step now, and again, I'm actively recruiting postdocs, so please let me know if your research interests fit to these directions. Thank you.
Wonderful talk. Amazing. I know. I listened to your talk, one of the conference, I believe, but again, this is amazing again. So
Thank you.
I want to say, if you're looking for a cool lab, with the amazing science, apply right now. So that's what I want to say. So let me start with the question. So the question is, this is a very important topic, especially we are talking about cancer. And then, as you know, the government, private, foundation basically invest a lot of money last several decades, but we're still talking about cancer treatment because we still need to develop, something, that cure the cancer. So just tell us or give us your insight why, after many decades of cancer treatment development, investment, we still have the problem, and then what would be the remaining challenges in the future to conquer the cancer? So that's my deep or very broad question.
Yeah, so it's an excellent question, and I'm kind of junior investigator, so my opinion doesn't carry much weight, but in my opinion, so cancer is kind of the part of your body, but with abnormal growth, right? So it takes a lot of longer time that this kind of small, because every single cells have mutations, they end up dying, or they're treated, they're kind of inhibited by immune cells, but some populations survive and evolve. So it's really hard to target that specifically. So it's different from COVID vaccine. So because it's a different pathogens, we can target it specifically, but tumors is part of our body, and throughout your life, many different mutations are evolved. So it's very heterogeneous, and it's very hard to specifically target. So that's why we are still suffering from this cancer. And especially as our life expectancy increases further and further, we are naturally exposed to more occurrence of cancers. Yeah, so I believe that our investment has kind of fruits already. Still some, but some population of patients have benefit from immunotherapy, or also colonoscopy prevents lots of occurrence of colon cancers. And we know that lung cancer everyone knows that smoking is bad, so we are actively doing social activities to decrease the smoking, which directly lowers the occurrence of lung cancers. I think our investment has do some jobs, but I think just cancer is really hard to target.
I see. That makes sense, because that means, my understanding about cancer is probably a little bit correct, because I believe the most challenging part is the differentiating between normal cell and cancer cell, and then that's why even some cancer treatment, some side effect, including hair loss, and so on. So that is part of the non-specific targeting, I guess. Am I right?
Right. Yeah. So
I see.
So far, one of the most effective drugs is killing all the cells with active proliferation.
I see.
Yeah. So cancer cells grow fast, and our health cells grow fast. So targeting all the highly proliferating cells. Actually, unfortunately, that's the most one of the most effective approaches. Yeah, but of course, there are side effects we have to.
I see.
Yeah.
That is also, you mentioned very important point, that is people do not realize, or many people do not realize, because of our effort to conquer cancer, we now have multiple different therapy, and that basically prolong our life expectancy even after we diagnosed by the whatever cancer. And then, as you mentioned, our life expectancy kind of increased, so that means we have the more cancer patient we probably do not see in the past, because people just die without any diagnosis. And so that's probably the reason, another reason we have more cancer patient rather than less, I guess.
Yeah, I think because of technology development and our investment, like for pediatric patients, their survival rate is over 90%. Of certain, like of course, some pediatric patients with cancer still have dismal prognosis, but most of the pediatric cancers have very good prognosis, because those kind of cancers are very specific to certain mutations, etc. So it's a bit relatively easier to target. But the other cancers that occurs throughout our lifespan is more heterogeneous. And yeah, and also there are some available therapies, but whether patients can endure the cytotoxicity is also another issue.
I see. That's very interesting. And my second question, while waiting, some question from audience, my second question is, what would be remaining challenges in probiotic engineering? You more focus on the cancer thing, but probiotic engineering, that's my favorite topic, so I want to ask that question anyway.
Yeah, so far, still some people still argue that the benefit of probiotics, some people it's not clear, and do we really need to have it, or etc. But I believe there's definitely some benefits, especially for targeting certain diseases. But it's really hard to, well, I don't know how to say, but like microbiome is super heterogeneous. It's really complicated.
Absolutely.
Yeah, so seeing the direct intervention effect with probiotics, that's why it's hard. So like some people, for cancer patients, they just wipe out all the microbiome and deliver specific probiotics or microbiome for treatment. But I think it's hard to, first of all, it's hard to monitor. There's too many strains there. So heterogeneity is a big issue. But I think like during this kind of, over the last 10 years, people have developed lots of technologies like big data, and I think we are getting there and there.
Sure. I mean, it's probably, I mean, cancer, I would say, we will probably, I hope, we conquer cancer in next 5 years or something like that. But probiotic, you also mentioned differentiating between, normal cell and cancer cell. That's very challenging, but that's basically two cell problem. One type of the normal cell, one type is the normal cell, the other is the cancer cell. But probiotic, we have billions of different, organisms in the gut, so they're probably much more challenging problem, I guess. So that's why I'm very interested, because that's challenging problem. I'm tenure professor. I want to do something crazy anyway. So that's why we also developed the consortium engineering tool, as you mentioned, specific targeting at the strain level.
I briefly mentioned that we talked about the E-cadherin. So the E-cadherin is kind of cell-to-cell junctions, and people have shown that, especially colon cancers, there's leakages there, and then E-cadherin is not it's dysfunctional in colon cancer parts. So we are trying to expand it from breast cancer to colon cancer, E-cadherin parts. So our goal in the few 10 years from now, probably the probiotics can generate certain materials which can activate E-cadherin so that the colon cancer or the epithelial layer of colons, they can make tight junctions between the cells or better junctions between cells, so minimize like inflammations or minimize tumor growth, etc.
That's wonderful. I believe you. And then please solve the problem for us within 5 years. 10 years too long to me.
Okay.
5 years. Okay, wonderful. So let me see. Let me check whether we have the question from the audience. Okay. In that case, let's close. In that way, I can go back to conference. One of the, actually that's one of the best, conference in synthetic biology. Many people are attending that conference at this moment, actually. So, let me close. So thank you all for joining and staying today, despite, as I mentioned earlier, the SYD conference happening at this moment. Actually, today we compete with keynote speaker. I think she now finished her talk or almost finished. We basically compete with her keynote talk in the one of the best synthetic biology conference in the world right now. But Sangmoo did a great job today. So
Thank you.
We'll meet again on June 8th, Thursday, the same time, the same Zoom link. We'll have Professor Scott Benter, the Chemical Engineering Chair of Columbia University, and Professor George Marchand at University of Washington. As usual, the follow-up informal chat will occur without recording. Please stay here if you are interested in chatting with us. I will promote you to panelists who can speak and show your face and your face basically if you wish. Thanks, and I'll stop recording. Thank you. Just give me 1 second.
Thank you.